because everybody leaves. Awesome. So today it's a real pleasure to get to introduce Dr. Eric Porges, who is not only a leader in the field, but he's also a longtime colleague and friend. I've known Eric since our time at UIC in the early 2000s, maybe, and I've had the opportunity to really watch his work and career evolve over the years. He's an associate professor in the Department of Clinical and Health Psychology at the University of Florida, where he also serves as the associate director of the Center for Cognitive Aging and Memory. And he's an assistant director of the Southern HIV and Alcohol Research Center. I think it's SHARC. And he earned his PhD at University of Chicago. So from a research perspective, he's been really focused on age-related changes in cognition and neurophysiological processes that underlie and accelerate these changes. His work has been particularly impactful in examining factors that contribute to accelerated aging, including HIV, heavy alcohol use, and obesity. And what's nice is that to address these questions, he uses a range of approaches that really span neuroimaging, including magnetic resonance spectroscopy, or MRS, cognitive and behavioral assessments, metabolic methods, and also neuromodulation techniques, such as transcutaneous vagus nerve stimulation. So please join me in welcoming Dr. Eric Porges and look forward to your talk.
All right, thank you very much. So today I'll principally be talking about our magnetic resonance spectroscopy work. So first, I'll give a little introduction about the methods, because it tends to be the case. If we were in person, I'd ask for a show of hands, but it's a relatively less widely deployed neuroimaging method. Then I'll talk about some of the work that we've done using this method to look at inflammation-associated neurometabolites, as well as their response to intervention. And then our work using this method to look at GABA, which is a lower concentration molecule in the brain across the healthy lifespan. So first, MRS is collected generally in the same scanner you'd use for research neuroimaging. We'll be discussing exclusively 3 Tesla data, though it has been deployed at 7 and higher fields as well, and occasionally at lower fields. So just to orient folks, if the blue oval represented, roughly average brain, an fMRI voxel, or the unit of measurement, you can think of it like a cube, is commonly 3 millimeters, isotropic 3 millimeter cube. A structural image might be 1 millimeter cubed, and MRS voxel, there is some latitude in this, but commonly, and much of the work I'll be showing here, has a voxel of 3 centimeters cubed or around there, so maybe 27 cubic centimeters, quite a bit larger. And as a result, some of the inferences we make and the study design approaches are different than you might have in the context of other neuroimaging. Here's an example of a voxel placed in a medial frontal position. Within spectroscopy, there are a wide range of approaches. I'm only going to be talking about proton spectroscopy, which is the most commonly deployed type of spectroscopy in human research. It uses, again, the same equipment we'd use for most other neuroimaging approaches. It's non-invasive. It allows us to quantify neurometabolites in vivo. And again, the spatial resolution is limited. So on the left is a typical spectra we might collect in a healthy individual. You can see a few peaks. You can see NAA, N-acetyl aspartate, GLX, which is principally a glutamate peak, but there's a glutamine and maybe a small amount of GABA contributing to it. There's a creatine peak, a choline peak, a myo-inositol peak, and then again, some additional creatine peak that's visible and labeled here. We'll be focusing on myo-inositol and choline. I'll describe a bit more about those metabolites in a moment. The way we quantify this measure, we take the spectra, and then we use what's called generally a linear combination approach, where we have models of the expected metabolites, and they're scaled linearly and then summed to match the data that we've collected. There are some alternative approaches, but this is by far the most widely used approach. The size of the signal is going to be proportional to the concentration of the metabolites. So as these peaks increase and decrease in size, that's reflective of greater and lesser concentrations. MRS has a longer history in clinical applications. Not the focus of this talk, but just as an example, in multiple sclerosis, MRS has been used, and when there's evidence of an elevated lactate signal, which is not normally visible in a healthy brain, it's evidence of a shift to anaerobic metabolism, perhaps mitochondrial dysfunction, inflammation. So it can tell you if there's active metabolic processes underway. Again, myoinositol, which will be one of the early topics in this talk, has long been proposed as a glial cell marker. It's found not exclusively, but predominantly in glial cells, or at least to a greater concentration. As glial cells become more active, their volume increases, and you see as a result elevated myoinositol. And this has been taken as evidence of increased glial cell activity, which you would expect in the context of an inflammatory response. It's been reported to be elevated in Alzheimer's disease and Huntington's, HIV, a wide range of populations where you'd expect neuroinflammation to occur. You'll hear me using terms like inflammation associated. It is one that is sensitive to inflammation, but could be argued to not be fully specific to inflammation. So I'm a little careful with the language. Choline, similarly, is a marker of membrane turnover, is associated with neuroinflammation. It is also more concentrated in glial cells, and quite a long history of research showing it increasing in the context of conditions where we'd expect inflammation to occur. It's been reported to be elevated again in multiple sclerosis, HIV, schizophrenia. There is, I think, some work now in MCI and HIV as well, though sometimes those results are a bit more complicated. A lot of the work that we've done has been around describing the age-related time course of these compounds and others. And a lot of our work both looks at normal aging trajectories as well as thank you, Leah, for the introduction, contexts where it's accelerated, both because the populations where we find accelerated aging phenotypes are important in their own right, but also they can inform our understanding of normal aging trajectories as well. So to introduce HIV in this context, and also as a aging relevant topic, over 50% of people living with HIV are over 50 years of age now. That cohort is continuing to age. I just updated this slide, and the number creeps up a little bit each time. And people living with HIV experience elevated risk for a number of conditions earlier than healthy populations. I'm going to focus on cognitive impairment here because we're talking about the nervous system, but cardiovascular disease, liver disease, diabetes, kidney disease, not a comprehensive list. And this has been discussed as accelerated aging, though some people have argued it might be accentuated aging. For this presentation, I don't think it's necessary to disentangle them, but there have been different ways of discussing this. And one of the things that we're very interested in the context of HIV and folks living with HIV is chronic inflammation impacting neuroinflammation, and through that, neurophysiological and cognitive consequences. So HIV and inflammation in the nervous system is a bit of a complicated story, much of it indirect, but not through the virus itself acting on neurons, but through other pathways, both systemically to the CNS and directly causing neuroinflammation and associated neurophysiological consequences. The literature goes back pretty far. So Linda Chang had some work over 20 years ago now, showing increases in choline and myoinositol in folks with HIV and in AIDS dementia complex, some evidence of maybe a further increase. And she argued, and I think this is largely supported by results that have come in the interim, that this is at least a partial reflection of increased inflammatory processes happening in these populations. compared to within her same work, this is again sort of foundational in this space. They also looked in healthy controls and showed age-related increases in both myoinositol and choline as well. So when we talk about inflammation, of course, many folks are going to ask, well, do these correspond with circulating cytokines? We have, as Leah also mentioned, quite a bit of work looking in obesity and specifically in populations who are receiving bariatric surgery. So looking at a cohort of folks who are receiving bariatric surgery, 52 to 89 years of age, cognitively intact. We collected a parietal MRS voxel, and we found in those populations that, yes, as other folks have found, that myoinositol and choline both increase with age, but when we control for age, the association between myoinositol, choline, and circulating pro-inflammatory cytokines were, well, actually, I should say cytokines generally were significant. And it suggests that the age-related impact on myoinositol and choline likely has, again, this sort of inflammatory, perhaps systemic to CNS pathway being engaged. There have been multiple reports of aging-associated increases in myoinositol and choline. Many of the studies have some limitations. They're often cross-sectional. They're often looking at disease versus control. Sometimes they compare young cohorts versus old cohorts, but they're not looking at a continuous population. And very often the age range is quite restricted. So working with a group at Hopkins led by Richard Eden, we've collected a cohort of normal aging folks from 20 to 60 years of age. This is about evenly collected between Hopkins and the University of Florida. We collected spectroscopy in this population in a white matter heavy voxel and in a gray matter heavy voxel. Here's an example of our spectra. And what we found is that both creatine and choline in both white matter and gray matter showed some evidence of an age-related increase. Now, this cohort's not terribly old, but they certainly are overlapping with the early aging. Interestingly, in this cohort, and it's sort of outside of the scope of this talk, N-acetyl aspartate, which is previously one that had been described as decreasing in aging, it's thought of as a neuronal marker, didn't show a change. Now, it's very possible that if we had looked farther out, this story may have changed. So building on that, we've been interested in seeing if we can push these neurometabolites around. So I introduced HIV earlier. We ran a study. The PIs of the overall study were Ron Cohen and Bob Cook, where we took folks with and without HIV who were heavy drinkers, and we asked them if they would engage in a 30-day contingency management study. So the point of the study here was to see if by dramatically reducing drinking, not as a test of the efficacy of the intervention, but taking an intervention that dramatically reduces drinking, can we impact evidence in the brain of neuroinflammation? Alcohol, of course, in addition to and perhaps interacting with HIV, can induce neuroinflammatory consequences. It's directly injurious to the CNS. Oxidative stress is a pathway that's commonly engaged through multiple mechanisms. Excitotoxic injury seems to be some evidence in the periods after heavy drinking and during cessation for very heavy drinking individuals, that there's a particular vulnerability. There's immune modulation. Of course, the HPA axis is engaged, and there's a whole gut-brain component that we're actively engaging. Unfortunately, that data is currently blinded, so I won't be sharing it here in this space as well. Previous literature looking at the relationship between myoinositol and choline, these inflammation-associated neurometabolites in response to alcohol, has been a bit complicated. So this study was an acute administration in rodents, and you used the same method. One of the nice things about spectroscopy is that we can use it in both rodents and humans using very similar methods. You don't have to sack the rodent. There's no tracer that's involved. So particularly in the context of longitudinal studies, it has some substantial advantages, as well as both for implementation, but also translationally. Choline in that population, or in the rodent cohort in the rodents did increase. Myoinositol story was a bit complicated, but was definitely modulated. It is worth identifying here that the measurements were taken pre and then 16 weeks out after alcohol. So the sort of time course in the earlier exposure is a little less clear. In the human literature, there was a recent review. The story is a bit complicated, but for reasons similar to the rodent study, when people are looking relative to alcohol detoxification or alcohol cessation is probably very important. And many of these studies get a baseline measure after detoxification, or the measure of detoxification is a number of weeks out. So the story is at the moment a bit unclear in that space. Our study, again, the 30-day challenge, involved a 30-day contingency management study. One of the interesting parts of this is that we did use biosensors. So we used a transdermal alcohol sensor. You'll see it here in the center on someone's ankle. We are also using devices that are a wrist-worn one outside of this study, more acceptable to populations generally. They look like a Fitbit. In the study, people with HIV, as well as heavy drinking folks who didn't have HIV, were recruited. We collected baseline data before they stopped drinking, and then they engaged in a motivational interview session, as well as 30 days of contingency management. If you're not familiar with the intervention, in concept, it's very simple. Essentially, they are paid not to drink, and that was verified principally through biosensor data. So we have a fairly high degree of confidence that drinking did or did not occur. We showed substantial reduction in alcohol consumption in these populations. These are self-report measures, and you'll see that folks reported extremely heavy drinking in the period before intervention. There's some heterogeneity in response to the intervention. We showed for myoinositol a reduction in marginal means in both people with and without HIV, and there wasn't a difference between these populations. So we have some evidence that myoinositol is reducing in response to a 30-day alcohol intervention, and the same with choline. So we found, I think, some reasonable evidence, admittedly quite a bit of heterogeneity in there, that a 30-day reduction in alcohol in very heavy drinking populations reduces these neuroinflammation-associated neurometabolites. But as I indicated, there is a lot of heterogeneity at the individual level, and the story is complicated there. And there's a lot of work. I know that Graham Mason up at Yale and Nora Volkov in NIDA has done, which I think suggests that there might be quite a bit of metabolic disruption, especially early in alcohol cessation, and that may explain some of the individual differences that we're seeing, and that's one of the areas that we're currently interrogating. So again, we see evidence after 30-day contingency management intervention that there is a reduction in choline and myoinositol, quite a bit of individual variation, but a reduction in marginal means. And again, HIV did not change these relationships at baseline or to intervention, and everyone was medicated and was virally suppressed in these cohorts. So now moving on to our work in bariatric surgery. So bariatric surgery and obesity generally have been implicated in neuroinflammation through systemic inflammatory pathways, blood sugar and insulin regulation, and quite a few other pathways. There was a recent paper making a pretty compelling case that the inflammatory consequences, this wasn't brain specific, but that the inflammatory consequences are downstream from the metabolic consequences. So that in response to bariatric surgery, the metabolic regulation has to occur before the changes in inflammatory processes. Looking at myo-inositol and choline in our bariatric surgery population, this is folks, you can see the BMI was 46 on average. Folks were 45 years on average, but quite a range of ages. We collected data before surgery and then again 1 year after surgery. This is a frontal acquisition. And we found that myo-inositol and choline do both decrease 1 year after surgery, and that the changes in choline were associated with change in body weight, and the change in myo-inositol were associated with change in HbA1c, which is a marker of average blood sugar. The related relationships, so body weight on myo-inositol and HbA1c and choline were very similar, but they didn't reach significance. So in response to bariatric surgery, both frontal myo-inositol and choline are reduced. Choline's associated with weight loss. We saw evidence that myo-inositol was associated with blood sugar reduction. Now, both myo-inositol and choline are increased. This is the evidence we showed earlier during normal aging. They often increase in the context of chronic disease, and they are responsive to interventions that target or are presumed to target neuroinflammation. Moving on to some of our work looking at GABA across the healthy lifespan. So folks here are probably familiar with GABA, but it's the principal inhibitory neurotransmitter in the central nervous system. There are both ionotropic GABA A and metabotropic GABA B receptors. It's involved in inhibitory signaling, excitation and inhibition balance, EI balance. And the metabolic context for discussing these GABA, glutamate, glutamine, they all share a common metabolic pathway, the glutamate, GABA, glutamine cycle. And that's relevant here because GABA is synthesized from glutamate via GAD, which there are two isoforms, both GAD67 and GAD65. Now, when we're talking about this measure, it's important to understand what it's reflecting, especially when we're discussing neurotransmitters. There's an impulse, certainly when I had many years ago when I first became involved with the method, to want to understand it as GABA being used in the context of neurotransmission. And I think that the field would argue, and I would, that we would probably need to substantially qualify that framing. GABA in the brain exists in multiple pools. So there is a cytosolic, a vesicular, and a synaptic pool coarsely to bind them. And the measure can't distinguish between these. The measure that we're going to describe here today is thought to largely reflect cytoplasmic GABA, of which GAD67 is likely, we shouldn't say like, is responsible for the majority of cytosolic GABA. So the implications for interpreting the signal, it's not a direct marker of phasic inhibitory activity. It's not a direct marker of, or an indice of GABA crossing the synapse or agonizing GABA receptors, but total GABA within the region we're looking at. So a framework that is often taken as one of sort of GABAergic tone or level of tonic inhibition. We think that the signal is largely from GAD67 being the primary source of cytosolic GABA, but it is worth sort of mentioning that GAD67 is not only responsible for cytosolic GABA. There is presynaptic GAD67 that's co-localized with GAD65, and So just an important qualifier on that. So when we're measuring GABA, and one of the reasons that this is a relatively newer approach and relatively less widely deployed is that GABA has a much lower concentration in the brain, maybe an order of magnitude lower than the metabolites I discussed earlier. So about a tenth of what you'd see for creatine, choline, and acetyl aspartate. And then on top of that, the spectra overlaps with these more plentiful metabolites. So the portion of GABA that we quantified generally is behind or overlapping with the creatine peak, making it difficult, perhaps impossible, depends who you discuss that with, to quantify without editing at 3 Tesla. It has a resonance that is very similar again to glutamate and glutamine, which is owing in part to its shared similar structure and common metabolic pathway. I'll point out here that when we are trying to quantify it, and I'll describe this more in the next slides, we take advantage of one of the protons that is at about 1.9 parts per million on this x-axis to deploy an editing pulse. And I'll describe more what that means, but there's an approach called megapress, broadly under this J editing family of spectroscopy approaches that allows us to quantify GABA and some other low concentration neurometabolites. The approach starts by collecting what we'll call off spectra, which is essentially the same as the unedited spectra I shared earlier and what we quantify choline, myoinositol, and other more plentiful metabolites with. We then apply an editing pulse at about 1.9 parts per million, and that refocuses the GABA signal, but you can think of it as largely nulling GABA's contribution to the spectra. It has some other consequences. You'll notice that this NAA peak is no longer visible because the editing pulse overlaps with that as well. We then take the original spectra, the unedited spectra, off, as well as the on with the editing pulse applied, and subtract them. And what that reveals by subtracting off where the GABA signal still resides, but obscured by creatine, and the on spectra where the GABA signal has now been largely nulled and the creatine peak remains. When they're subtracted, we get, it's called a different spectra, but we have our GABA peak, and that's what we use to quantify GABA in spectroscopy. You'll notice MM here, that refers to macromolecules. There are some other compounds that reside within this peak. There are some approaches to remove those, though generally results have been highly correspondent whether or not you're removing macromolecules or not. There are some substantial challenges that occur when approaches are taken to try and reduce their contribution. When we're collecting GABA spectroscopy or other edited approaches, we have to use quite a large voxel. Again, 27 cubic centimeters is common. There's a little bit of latitude to go smaller or larger, and long acquisitions are really important. So the consequences of using a voxel that might be half the size, for example, would require 4 times as long an acquisition with the way that the signal scales. But generally, it's difficult to get these acquisitions much below 10 minutes, which makes it incumbent upon the researcher to have a strong a priori prediction about where they're interested in looking. Again, this is in contrast to other MRI approaches where you can generally collect whole brain acquisitions, and limits the total number of areas that we can collect. The signal has been within subject designs shown to be stable diurnally, which is very helpful, of course, when you're designing a study, that we don't have to be too considerate about that, at least with the GABA signal that we're looking at. And we were recently part of a collaborative multi-site, international multi-site study looking at the stability of GABA across the menstrual cycle. And we didn't see any evidence in this study that there were systematic changes across the menstrual cycle. So again, how do we interpret GABA concentrations? Overall GABA levels are therefore a better marker of GABAergic tone, i.e. levels of tonic inhibition than inhibitory activity per se. In our work and others, in general, we've seen higher concentrations are associated with better cognition. I think it's very plausible that at extremes you might see an inverse U-shaped relationship, but within the cohorts we see, we generally see an increasing GABA concentration related to better function, and that is largely in the context of a structure-function relationship. And I'll share a couple examples from the literature describing that in a moment. This was a review from Stefan Swinen's group, citing some of our work and other groups, trying to organize the GABA literature. And they make an argument that higher GABA, at least as quantified using this measure, is associated with an increased distinctiveness of neural representation. So facilitating the suppression of noise and the enhancement of signal noise ratio in neural representations that may result in a better ability to cope with interference. And there's also quite a bit of evidence, and this maps on well to some of the rodent work with the suppression of prepotent responses. Some early work in visual processing, higher GABA concentrations in the frontal eye fields, which are involved in the volitional control of eye movements, were associated with a reduced distractor effect. So people were essentially they were better performing in the context of an eye movement task where they were told not to move to certain targets as GABA concentrations in the frontal eye fields increased. In a wide range of sensory processing, there have been similar patterns of results from Nick Putz's group, and he's quite prolific in the autism space using these methods, has shown that tactile sensory discrimination in the sensory motor cortex better performance is associated with higher GABA concentrations, but that relationship didn't hold in other regions of the brain. Again, sort of arguing for a fairly specific consideration of the tissue that supports the capacity being related to performance on it and not necessarily a global GABA concentration. Presbycusis, or age-related hearing loss. We have a project going on in this space as well. Higher GABA concentrations were associated with a better ability to distinguish pure tone averages. So they were more sensitive to distinguishing tones when GABA concentrations in auditory cortex were higher. Clinical populations have been explored. So there's evidence for reduced GABA in MCI. There's been reports in AD that follow similar patterns. In the MCI population, the performance on a number of cognitive tasks was again associated with the GABA measurement, where higher levels were associated with better performance. This is a backward digit span task. In alcohol, which is an area that we're actively exploring, and we have some papers that hopefully will be in press soon. The story is a bit complicated. I think this is a similar story to what we've seen with myoinositol choline and inflammation-associated metabolites, where it's probably pretty important to look at where someone is, either in active use or detox. And GABA may be even more complicated than myoinositol and choline, given alcohol is a allosteric modulator of GABA A receptors. The time course of response is likely important over the hours following the most recent drinking event. And this has, at least in the historic literature, not been aggressively attended to, rather treating it as a trait measure. In psychiatric disorders, there's some evidence that GABA is reduced in meta-analysis across all disorders, though depression may be driving this. From Simonet's group up at University of Maryland, there was a fairly recent meta-analysis, and they made pretty compelling case that, at least in the rostral medial frontal cortex, the GABA is reduced. And interestingly, though it's a smaller pool of data, when depression is in remission, that GABA looks normal. Moving into our aging work, there is extensive rodent literature showing GABAergic alterations. Much of it's using GAD labeling, and GAD certainly changes throughout the brain during aging, and there are cortex layer level differentiation in that response. But both GAD 65 and 67 are impacted by aging. Quite a bit of regional variation. Much less data in humans. This is a post-mortem study. It's important to note on the x-axis that this is not a linear scale, and that this first index is 1 year, 10 years, and 80 years. But there is evidence for cortical GAD 65 increasing, perhaps relatively stable in early adulthood, and then decreasing over the lifespan. Prior to the work that we did, there had been one study looking cross-sectionally across an age range that included early aging, and they showed both in frontal and parietal regions reduction in cortical GABA. So we sought to extend this work out, collecting an older cohort, and we found that GABA decreased both in frontal and posterior regions, of parietal voxel. They're visualized up top. I would focus first on the red line and say that this is our measurement of GABA concentrations, and that the blue and green line show post-processing approaches that attempt to address atrophy. Because the MRS approach collects total GABA within the voxel, depending on the analytic method you're using, if you're in a context where you'd expect atrophy and there to be less brain tissue as a result, you will see a reduction even if the tissue within there has the same concentration of GABA. These approaches on the right attempt to address that using a couple different approaches outside of the scope of this talk. Tad Polk's group recently did a longitudinal study, and they showed a similar pattern. So this isn't simply demonstrated in a cross-sectional cohort, but there's evidence that longitudinally across a number of measurement sites within the brain, the GABA does decrease. Again, they're using a couple different approaches to address for atrophy. This is one by Ashley Harris's group called an alpha correction, which is a fairly aggressive one that assumes a 2-to-1 ratio of GABA in gray versus white matter, also accounts for atrophy and some other relaxometry related or considerations, that this relationship is still preserved. We also looked at the relationship earlier on in our work between GABA and cognition, and what we found is that GABA in a frontal voxel was related to cognitive performance. On the x-axis is the MoCA score. This is a pretty coarse cognitive screener, but we showed a relationship between frontal GABA and cognitive function, and that relationship was not impacted by tissue correction. So again, our attempts to address atrophy and less brain tissue overall didn't change this relationship. There wasn't a relationship in our posterior voxel. So reports describing changes in GABA from our work and some of the work I've showed here seem to indicate that during aging that GABA decreases. However, there has been some I'm reluctant to call them conflicting results, but some reports in literature that had made statements like GABA increases during aging, that it's stable during aging, or stable with age anyway. And we sought to better understand that. And one of the frameworks that we were sort of engaging with when dealing with that was, people may be familiar with this sort of parable about, in this case, it's blind scientist and an elephant. It very much depends where you're looking as to what inferences you might make about what you're interrogating. So as I described before, there had been some lifespan data that made a case that at least GABAergic function both increases early in life, is stable-ish during midlife, and then decreases during aging. We engaged in a meta-analysis where we looked at any published results that had used the megapress method for GABA quantification. It's probably worth saying that there are some studies that have used other approaches at 3T, especially some older papers, and I think that largely the field is a little skeptical of some of those approaches. I think it's broadly understood that it's difficult without using an approach like this to quantify GABA at least at 3 Tesla. These were the papers that we included in the meta-analysis. And this was not a conventional meta-analysis, but rather an individual participant data meta-analysis. And we extracted data from any published scatter plot that had individual data. So we used a tool that let us identify data points and estimate their value based on the scaling of the X and the Y axis. And from that, as well as adding some data that we had collected or collaborators on the project had collected, we were able to get reasonable coverage across the lifespan. Here are the linear fits for the respective studies that we integrated into this. Again, thinking about this sort of depends where you're looking framework. As we plot our data, we can see maybe some sort of image emerging from the aggregated results. But merging the data is challenging because the unit of measurement is not consistent across these studies. There are different ways of quantifying it. They are not perfectly compatible, and they're scaled differently, but they're largely compatible, though the scaling is different. So we used an approach implemented in the STAN package that was based on what had been done with political polling. So we attempted to generate what's sometimes called a house effect, or the systematic bias of the individual studies, and then merge the data accounting for that. Here is the product of that analysis. This is with quite a flexible spline, but we show some evidence of an early increase, stability until maybe the early 40s, and then a gradual decrease during normal aging. And again, this is all with normal aging and cognitively intact populations. It fit reasonably well with a log normal model. And I would say, again, noting the x-axis, it may have some similarities to what's been seen in the pathology literature. We then extended this into the oldest old. We, as a Center of Cognitive Aging, have been involved in a multi-site study where we're collecting cognitively intact folks who are 85 and older. This is called the McKnight Brain Aging Registry. And that data set, by the way, is available if people are interested. It was a study of cognition and brain function in the oldest old, but again, importantly, cognitively intact oldest old individuals. There's about 200 participants that were recruited overall. Not all had all measures across the 4 sites that were supported by the McKnight Brain Research Foundation. So that's University of Alabama, Arizona, Florida, and Miami. 100 individuals had spectroscopy data. They range between 85 and 99 years. And again, they were cognitively intact. There was a single voxel collected in a frontal region. This is using the same method we described earlier. Here's a quick example of the spectra collected across the sites. Through integrating these data, and this is now published in Translational Psychiatry, led by my grad student, who's now a postdoc in Graham Mason's lab up at Yale. Through the inclusion of these 85 and older adults, we see some evidence that the velocity of change starts to be impacted around 80 years of old. And this is likely the reflective inclusion of a large number of cognitively intact older adults. But of course, if you are going to collect in populations out at this extent of the age range, there is a big survivorship effect, right? So while they may be intact and perhaps described occasionally as normal or healthy, the average individual at 85, 90, 95 is not cognitively intact and is no longer, at least in the US, even alive. So noticing here, this orange bar, we see that the velocity of change between 80 and 90 or so decelerates. So these are a highly selected atypical minority of their both birth cohort to make it both to be cognitively intact at this age and even to be alive. Again, the life expectancy in the US is 77 years of age. And then amongst this population, the estimated prevalence of MCI and dementia is substantial. So during typical aging, you have accumulating physiological changes, and GABA seems to be one of them. And if we were to follow out the sort of inferred trajectory from the younger aging cohort, we would likely be putting people into a range that's not compatible with normal cognitive function, or the physiology from which the cognitive function emerges in their CNS would no longer be able to support that if GABA were to continue to decrease. And I'll make a point that this is probably a generalizable consideration. We're looking at GABA here, though. So when the physiological substrates from which cognition emerges have a lower limit, they likely have a lower limit necessary to support performance. And this has been sort of conceptualized by groups as something of a functional threshold, not in the context of GABA or neurometabolites, but that when you have a functional threshold, at that point, an individual would transition into a dysfunctional state. So they would no longer be eligible in a study where you had some threshold demonstrating, that they were cognitively intact or normal or healthy. In this work, they were focused principally on independent living, but I think that it's readily generalized to cognitive function in our context. We here have the model from our spline fit from the data I showed earlier, somewhat simplified and imposed upon their data set. And what we're arguing here is that in our cognitively intact oldest old adults from our non-clinical sample, we had an implicit floor for function that was tightly coupled to the physiology from which it emerges. And if we're thresholding inclusion based on that, or if we're discussing independence or any other capacity, then you're going to have physiological consequences that parallel that. I want to thank everyone who's been involved in the research. I'm eager to take questions, and I certainly want to acknowledge the institutes that have funded this work and much of our ongoing work. So thank you very much.
Thank you, Eric. It's an amazing body of work that you've done. I'm gonna open it up to some questions. I see Jules and Tony. Do you want to unmute yourself, guys? We'll go one at a time. Jules, you had the first question.
Okay, thank you. Yeah, hi, thank you for a very detailed talk. There's a lot of information, so I have a few questions. Number one, what about people with HIV? I don't think I saw any data. I would expect that older aging people with HIV with comorbidities might have lower GABA, and without comorbidities might still have higher GABA. That's the first question I'd like you to comment on. And number two, and of course, how would that correlate with cognitive function in terms of screening for cognitive testing like MoCA and so forth, and maybe self-reporting by patients? And number two, what about injection drug use, do you have any data? Have you seen any data on people with a history of injection drug use, maybe 10, 20 years ago, they've stopped, and GABA levels? And what about current cannabis use and GABA levels? Is there any data that you have on that? Thank you.
Okay, a bunch of questions, all interesting and relevant to topics we're either well, many of which we're actively engaging. So first, in the context of HIV and aging, the work that I showed here and the cohorts that we've collected with HIV, where we've used this measure, those have been heavy drinkers, often very heavy drinkers. So from our data specifically, I'm a little reluctant to make inferences, but Linda Chang has done work in this space, and I think that there are both physiological reasons and I believe now some data in normal aging, very difficult to parse out comorbidities, but that HIV does impact GABAergic function. So Jay McLaughlin, who's here at the University of Florida, has done some TAT model work, which is, this actually feed into your next question, which is, definitionally, of course, HIV is a human disease, but you can induce HIV-related proteins in rodent models. And in his rodent models, this is not an aging question, but a substance use one, the induction of these proteins, TAT specifically, the work I'm thinking of now, you can increase place preference for both alcohol and I believe methamphetamine as well. And I assume that they were injecting the methamphetamine in these rodents. So the story between alcohol and substance use is one that I think historically was largely viewed through context in which people who had HIV were likely to live, stress upon them, other factors. But I think that there actually is some compelling evidence that HIV may, through TAT and perhaps other mechanisms, actually increase vulnerability to substance use. And in these place preference models, they provide alcohol or methamphetamine, and then they stop, and then they reintroduce them into the environment, induce the TAP protein, and the rodents go seeking, apparently craving both alcohol and methamphetamine is literature I'm familiar with. I would probably expect that to generalize beyond to other drugs, especially in the intravenous space. So I don't know about opiates. So the aging work, just to touch back on the earlier component, we are doing work in that space with less, there are still folks who are drinkers, but they're less heavy drinkers, where we are looking at differences in them, but I'm unfamiliar. I'm a little reluctant to make very strong statements here, in part because of the comorbidities you identified, variability in treatment regime. But my suspicion is that a very well-managed individual with HIV may show some evidence of altered GABA. We certainly have evidence of cognitive consequences. But I hope to report back from our own data in the not too distant future.
Thank you on that one. I know we're kind of coming down, but Tony?
Hey,
Hi.
trying to get my mic, trying to get unmuted there for a sec. Eric, great talk. I really enjoyed listening to that. So Jules had several of the HIV questions. So thanks for answering those. I had one question just about your discussion of what the GABA signal reflects. So I totally agree that it's basically everything in that voxel. Now that being said, like a bunch of us, so I'm at the Institute for Human Neuroscience in Omaha, and here we do a lot of spectroscopy now, mostly in collaboration with folks at Hopkins, who are really helping us on the technical pieces. But one of the things we argue about a lot is what the person should be doing. Like, okay, so it's everything in the voxel, but does it matter then if you're looking at the calcarine cortex and their eyes are closed, or they're watching a movie, or they're doing these different things? And the opinions are like, across the board just within our building. So do you know any papers that have examined that? And just what's your view on that?
Well, so functional MRS is a nascent field. I mean, it's been nascent for a while, but the one of the CVs, depending on the method you're using, are okay for this method. It is admittedly a kind of a noisy one, and there have been reports where, for example, under heavy cognitive demand, you can maybe show some decrease in GABA in a relevant area. Though I think folks have also had trouble replicating some of those. I think the field largely has not concerned itself a ton with behavior of the participants as long as they're still, which is more of a methodological than a physiological concern. Pharmacology definitely can impact it. So Graham Mason did a study where they used intravenous alcohol administration in healthy, sort of minimally drinking in their grad students, and they showed a very quick reduction in cortical GABA after the administration of it. I think there have been some similar findings with other GABAergic agents. We have done a little bit of internal explorations with oral alcohol administration. It hasn't been published, but showing things that may mirror what Graham had shown. It's worth pointing out that Graham Mason's work was intravenous alcohol, where they're able to hold a very precise blood alcohol concentration for the duration of it. I wish I had a better answer. My impression from the literature, in my opinion, is that it probably is less impactful than the between group or between condition, between intervention considerations people are interested in, except maybe at the extremes. And there's very little literature where people are really being pushed in the MRI and watching it dynamically.
So do you think, maybe just one quick follow-up, any kind of differences that are seen there? So like what the person's doing, do you think for maybe GABA and GLX, it more reflects like different sensitivities to parts of the metabolic pathway?
Yeah, so I mean, it's obviously containing information about metabolic pathways. It's containing information about GABAergic neurons, sort of their population health. And I think that as a framework to disentangle this entirely is something we're still kind of working towards. But it again, being responsive to GABAergic pharmacological interventions is suggestive of what it reflects. But its relative stability, for example, we showed the diurnal data within folks where within participant it's relatively stable over the course of a day, is suggestive that, and these people weren't going through an intervention, that at least the demands that someone would incur during the course of a normal day are unlikely to meaningfully impact it.
Awesome. Thank you. Excellent talk.
Thank you.
And I'm going to let Carl squeeze in one more. I know most people are jumping off, but I feel bad because Carl always comes up with the last minute question, and I've missed him a few times on a bunch of calls. So Carl, are you still there?
Yes, I'm here. And I was very interested in the talk and wondered specifically if you've looked at separating the GLX peak into a GLU and GLN as well as NAAG with respect to inflammation and aging.
Okay, so for the first one, there had been field-wide, I think, reluctance to take a straight ratio between GLX and GABA. We have done this recently, and I think there's been an increasing sort of tolerance for it, in part because of the larger metabolic role that glutamate may serve and some of the inferences that might produce. I didn't discuss it here, but there are different ways, of course, that glutamate can be quantified, right? So when we're editing for GABA, we co-edit a glutamate peak, largely a glutamate. There's a glutamine component. But glutamate, or the GLX peak anyway, is also evident in the unedited spectra. So the results don't always correspond perfectly, depending on how glutamate is quantified. Paul Mullins had done work a number of years ago and made a pretty compelling case that if you're going to use a conventional MRS approach, there are some very specialized like 2D approaches, but if using a conventional one that in 80 millisecond echo time, because of the way that the glutamine signal evolves, produces a fairly pure glutamate signal. One of the features one of the sort of consequences of that is that megapress and other related edited approaches, well, what I showed here is done at 68 milliseconds, can be done at 80 milliseconds. And we often do that So if you want a relatively pure glutamate signal and to look at that either independently or in the context of your edited GABA peak, that is there. I have seen, and we have also had some results where the glutamate and glutamine ratio the glutamate to GABA ratio does seem to be interesting. I didn't share it here, but we do have that same alcohol intervention study that I described. We are looking at changes in GABA in that population. We do see a very similar story for both GABA generally as a water reference metabolite and GABA versus glutamate. They show very similar findings, suggesting that largely what's changing in that context anyway is GABA and not glutamate. We also don't see a general glutamate change there. But this is an interesting and important question that I maybe there'll be a nice consensus paper about the appropriateness of the GABA GLX ratio and and how we interpret it. In terms of the NAAG comment, that is not something that at least I feel very comfortable from the data I showed here disentangling. However, there are edited approaches very similar to what we've done for GABA. If you're specifically interested in disentangling NAA and NAAG, that can be done. And we are, in people with HIV, collecting that data now. We are using an approach, this is from Richard Eden's group, it's called Hercules, which is a similar but kind of evolved editing scheme, which lets us look at multiple targets from the same acquisition. And we may be able to disentangle some of the NAA, NAAG signal in that data. And maybe I'll be able to get back to you. I as well am also interested in that. Of course, because of methodological limitations, the field has traditionally not concerned itself with distinguishing NAA from NAAG. But it is very interesting, and I think that finally the methods are now allowing us at 3T to do that. Of course, at 7T, higher fields, things become a bit different, but that's a relatively rare opportunity for us to deploy.
Thank you. Great talk.
Thank you.
After Barb's on here, I touch base with you later because she's very interested in NAG. So we've used secondary data, but it was with 7T and older folks with HIV has shown that higher NAG in brain regions will lead to better cognitive function. And then her group developed a approach to measure it in CSF, and just published that with our linking CSF NAG to a very specific cognitive domain. And then in a subsequent paper, kind of linked it to an inflammatory signature. So Barb's on here. I know those people. I'm supposed to go to a 1 o'clock, but I know this is an area of huge interest to Barb.
Yeah, I was going to ask questions, but we were running out of time, so maybe we can follow up afterwards.
Absolutely, yeah.
Okay.
Yeah.
Thank you guys for hanging on a little bit later. Caroline, who's speaking next week?
It is, so we have 2 talks next week. It's still an hour, but we're having 2 talks. Increasing NAAG restores cognitive deficit in a murine model of HIV, and that's Mei Huang from Johns Hopkins. And then we also have targeting gender-specific gut microbiota improves social and cognitive impairments, and Echo HIV infected mice by Yan and Lee, also at Johns Hopkins.
Yeah, so Eric.
I'm having trouble hearing you, Leah.
Yeah, I can't hear you either.
You were good earlier, but maybe you switched microphones.
Yeah, you're going in and out.
I don't know. But anyway, Eric, should send your folks here to come to these talks, because I think
there's
a lot of things. And I see Tony's on here who gave a talk. May want to, I don't know if we taped him, but he's another gave a great talk on his imaging. So on Meg. So thank you all. I'll be in touch anyway.
Yeah. Lee, I couldn't hear what you were saying about NAG, but I want to follow up with you about that. It was going in and out, but we'll touch base soon. Thank you all very much. It was a wonderful opportunity, and I will send some of our folks to these in the future. Thank you.
Thanks.
Thank you.